Guys, now let's continue with the different types of lipids in the cell membrane. See, in cell membrane, there are different types of lipids called as phospholipids, glycolipids and cholesterol. In the recent exam, the question was asked from the topic of the phospholipids. Okay, I will tell you what is that question also. In the recent exam, the question was asked. First of all, you should know what exactly is a phospholipid, what exactly is a glycolipid and cholesterol. See, phospholipid is nothing but a lipid. Let's understand. It's a lipid. It's some lipid to which some phosphate group is attached. Okay, phosphate group is attached. So phospholipid means a lipid to which a phosphate groups are attached. And what exactly is a glycolipid then? Glycolipid, sir. Glycolipid is also a lipid to which some carbohydrate group is attached. Okay, some carbohydrate group is attached. So that's called as a glycolipid. Now, you know, in our cell membrane, phospholipids are the major lipids. Phospholipids are major. Okay, already I have taught you our cell membranes are mainly made up of proteins. 55% of the cell membrane is made up of proteins, that's fine. Lipids are 45%. But out of all the lipids, what is a major lipid? The major lipid is phospholipid. That's why we call it as a phospholipid bilayer. Okay. Now, what are the different examples of the phospholipids? Same, the first type of phospholipid that I am teaching you is called as a lecithin. Lecithin. Second type is sphingomyelin. Now students, whenever they hear this lecithin and sphingomyelin, they will start to have a doubt. Sir, we have studied about this lecithin, sphingomyelin in the topic of the respiratory physiology. Lecithin and sphingomyelin, they are the major lipid and minor lipids present in the surfactant, which is helping to decrease the surface tension in the lung. See, yes, it's the same lecithin and sphingomyelin. Guys, remember this lecithin and sphingomyelin, they are not only present in your lungs, they are not only present in the surfactant, they are also present in each and every cell membrane. So each and every cell membrane, they are having this phospholipid called as a lecithin and sphingomyelin. So the take home point is lecithin and sphingomyelin, they are not only present in the surfactant, they are also present in each and every cell membrane. Okay, so the one is called as a major lipid, other is called as a minor lipid. Okay, in surfactant, lecithin is a major lipid and sphingomyelin is a minor lipid. Okay, next, the third type of phospholipid, I have told you in the recent exam this question was asked. This is the question. The third type of phospholipid is called as a phosphatidyl. Okay, phosphatidyl serine. You can call it as a PS, in the short form PS, phosphatidyl serine. See, this phosphatidyl serine, it is always internalized. What does I mean by it's always internalized? See, in a healthy cell, let's take a healthy cell. Okay, see, this is a healthy cell or happy cell, let's say. So in this healthy cell, always the phosphatidyl serine, this phospholipid, phosphatidyl serine, it is always internalized, which means it's always towards the cytoplasm. But when a cell is committing suicide, okay, when a cell is committing suicide means it's undergoing death, sir. So what is that process called as apoptosis? You know, when a cell is committing suicide, this phosphatidyl serine, now it will be externalized. It's going to show outside. It's going to show up outside. This is called as PS flipping. So the phosphatidyl serine is going to flip to the outside. Okay, now the cell is undergoing death. So why it is flipping outside? It is giving a signal. Okay, it's giving a signal to what? It is giving a signal to the macrophages. So macrophage, I am dying, you come and clear me, you come and eat me. So the cell which is dying, it is giving a signal, eat me signal. So this is eat me signal with the help of phosphatidyl serine to the macrophages, so that the macrophages will come and clear these apoptotic bodies. Okay, so now phosphatidyl serine, it is always internalized in the healthy cells, but it is getting externalized in the cells which are undergoing apoptosis. So now tell me how to identify apoptosing cells. In an apoptosing cell, the phosphatidyl serine flipping is seen. Now, why this is very much important for your exam? See, now I am using a dye which is called as annexin 5. Okay, now the dye that I am using is called as what? Annexin 5. See, this annexin 5, it will go and bind with phosphatidyl serine. It will go and bind with what? Phosphatidyl serine. So now tell me, if I am using annexin 5 on a healthy cell, if I am using it on a healthy cell, do you think the dye can go and bind with the phosphatidyl serine? No, the dye cannot go and bind with the phosphatidyl serine because the phosphatidyl serine is never exposed. Okay, it's always inside. So annexin 5 cannot bind with the normal healthy cells. But what about the annexin 5 with the apoptosing cells? Now this annexin 5, now the annexin 5, let's say, now this annexin 5, it can go and bind with what? It can go and bind with the PS, okay, phosphatidyl serine. Now whenever this binding is happening, whenever the annexin 5 binds with the phosphatidyl serine, it will give the lighting, it will give the fluorescence. Now with this fluorescence, you can identify the dying cells. Okay, by the fluorescence, you can say, this cell is dying. So now annexin 5 is considered as, because of this, annexin 5 is considered as what? It's a marker of apoptosis. Annexin 5, okay, let me write here. Annexin 5 is a marker of apoptosis. So you can identify the apoptosing cells by using a dye called as annexin 5. Done? Now, so successfully what we have completed, guys, we have completed the 3 types of lipids in the cell membrane. The first type of phospholipid is called as lecithin, sphingomyelin and phosphodiesterase. Now let's discuss the 4th type of phospholipid. The 4th type of phospholipid is called as a cardiolipin. Now where exactly this cardiolipin is present? See, in the name itself it is there, cardio. This question was also recently asked in the 2023 exam, I think. A cardiolipin. Cardio meaning heart. So the cardiolipin is exclusively present in, okay, present in mitochondrial membranes of the heart. Mitochondrial membranes of heart. Okay, it's exclusively present in the mitochondrial membranes of the heart, and this is the question asked. Cardiolipin, where is it present? In the mitochondria. This question was asked. Okay. Now, okay, sir, it's a phospholipid. But why this is important for your exam is because in certain diseases, okay, in certain systemic diseases, patients will produce antibodies against this cardiolipin. Patients are producing autoantibodies against this cardiolipin. So, right, anti-cardiolipin. See, anti-cardiolipin antibodies are seen in which diseases? Diseases like syphilis. Okay, syphilis is a sexually transmitted disease, because of the Treponema pallidum. Okay, spirochete. So syphilis, the persons who are suffering with syphilis. See, imagine I am the one who is suffering with syphilis, for example. I will produce antibodies against not the bacteria. Okay, against the bacteria I will produce antibodies, but I will also start to produce antibodies against the cardiolipin called as against the phospholipid called as a cardiolipin. Okay, so syphilis people will have anti-cardiolipin antibodies. Okay, and not only that, APLA syndrome. You know APLA syndrome. APLA stands for what? Anti-phospholipid antibody. See, anti-phospholipid. Which phospholipid? Cardiolipin is a phospholipid. So, anti-phospholipid antibodies are seen in APLA syndrome. So, these anti-phospholipid antibodies will cause the thrombosis in the placenta and will lead to the recurrent abortions in the females. Okay, so the females who are suffering with the APLA syndrome, later you will study this in your obstetrics. The person who is suffering with the APLA syndrome, because of these antibodies, there will be placental insufficiency, because these antibodies, they will go and cause the clots in the placenta, leading to placental insufficiency, leading to abortions. So a patient who is suffering with the APLA syndrome, just remember it. It will cause second trimester, okay, especially in the second trimester, recurrent abortions, again and again abortions. Okay, it's autoimmune disorder. So now tell me, there is a lipid called as cardiolipin. This cardiolipin is present where? Mitochondria, in the heart, okay, in the mitochondria of the heart. So anti-cardiolipin antibodies are seen in which diseases? Diseases like syphilis, which is a sexually transmitted disease, and an autoimmune condition called as APLA syndrome. Next, sir, after discussing about the phospholipids, these are the main phospholipids that I have discussed. There are also other phospholipids like phosphoridyl, inositol, okay, different things are there, but these are the main important things which I want you to know for your exam. Next, look at the second type of lipid. The second type of lipid is called as a glycolipid, okay, lipid to which some carbohydrate group is attached, glycolipid. Now, based on the structure, the glycolipids are further divided into types like cerebrocytes, gangliosides. And these gangliosides, they are also present in different locations like GM1 ganglioside, GM2 ganglioside, GM3 ganglioside. So, gangliosides, they are again of further different types, GM1, GM2, GM3. Okay. Now, one important thing is this GM1 ganglioside. Where exactly is this GM1 ganglioside present? Okay, where is it present, sir? So this GM1 ganglioside, it is present on your intestines. Let's say here is your intestine. Okay, here is your intestine. Now in your intestine, okay, let's say duodenum. Here is the GM1 ganglioside present. Look here. Here is a GM1 ganglioside. Now what is the GM1 ganglioside doing? Let me tell you. Imagine today you are having a ingestion of contaminated water. Okay, today you went outside and you want to try some street food today. So what kind of street food, like let's say the famous street food in India, let's say you want to try a pani puri or some golgappe. So what kind of water you are going to get? Definitely, is he going to use Bisleri water or what? No, definitely not. You are going to get the contaminated water. And let's imagine in that contaminated water, there is this Vibrio cholera present. So now you got cholera into your intestines. Okay, cholera entered into your intestine. What this cholera will do? This Vibrio cholera today, it is going to produce this cholera toxin. Okay, cholera, what it will do? It's going to produce a cholera toxin, guys. Now this cholera toxin, see, it is getting access into the cell. Okay, the cholera toxin, it is getting access into the cell by using this GM1 ganglioside. So this stupid GM1 ganglioside, what it is doing, it is acting like a gate. It's acting like a receptor for the entry of the cholera toxin into our cells. So okay, cholera toxin enter into the cells. You know what this cholera toxin will do? It's not going to sit calmly. This cholera toxin, it is going to activate some G proteins, and it's going to activate an enzyme called as adenylate cyclase. So adenylate cyclase enzyme is getting activated. Now once you know the adenylate cyclase is activated, secondary messengers are going to be produced. Which secondary messenger is getting produced? The cyclic AMP levels are going to increase. Now what happens if the in the intestinal epithelial cells, if the cyclic AMP is more, there is more cyclic AMP, the cyclic AMP, what it is going to do, it is going to cause the secretion of sodium and chloride. It's going to cause the secretion of the sodium chloride into the lumen of the intestines. So into the lumen of the intestines, more sodium chloride is getting secreted today. Okay, more sodium chloride is getting secreted today. And you know, the sodium chloride, wherever sodium chloride goes, water follows. So today, more sodium chloride and water is getting secreted. More sodium and chloride is getting secreted. More water is getting secreted into the intestines. So now, as more and more water is getting secreted, so the patient can have diarrhea. So this patient is going to suffer with diarrhea. You know, cholera is going to cause the diarrhea. The main problem with the cholera is diarrhea. No. So how the diarrhea is going to look like? So this diarrhea is going to look like rice water. Okay, rice water like diarrhea. So now why I'm telling all this is because how the questions are going to come up in your exam. Okay, because the questions will come from this area. What are the questions? Questions are, cholera toxin uses GM1 ganglioside. So tell me what exactly is this GM1 ganglioside? So GM1 ganglioside is nothing but a lipid. Which type of lipid? It's a glycolipid. Okay, it's a glycolipid. So GM1 ganglioside acts like a receptor. Highlight here, GM1 ganglioside acts as a receptor for cholera toxin. Now tell me, cholera toxin acts via the which secondary messenger pathway? Cholera toxin acts via the cyclic AMP. What kind of diarrhea is going to be seen? Rice water like. Okay, if you wash rice, how the water going to be like? White in color, no? So that's how the diarrhea is going to be like, you know, like a rice water. So that's why it's called as a rice watery diarrhea. Okay. Next, look at the third type of lipid in our cell membrane. The third type of lipid in our cell membrane is a cholesterol. Okay. Now I'm asking you, tell me, sir, do you want a cell membrane to be flexible or rigid? See, our cells has to grow. Okay. So through the cell membrane, there should be exocytosis, endocytosis. Okay. For all these processes, the cell membrane should be flexible. Okay. The cell membrane should be flexible, sir. So who is going to control that flexibility or fluidity? The answer is cholesterol. So cholesterol acts as fluidity buffer. Okay, it acts as fluidity buffer. It maintains the flexibility of the cell membranes. Okay. Now, okay, sir. In our cell membrane, there are three types of lipids present. But which type of lipid is never seen in the cell membrane? Never seen, sir. The answer will be triglycerides. So triglycerides are never ever seen in the cell membranes. Okay. So now just as a recap, tell me how many types of lipids are there, guys? There are three types of lipids: phospholipids, glycolipids, and cholesterol. Who is the fluidity buffer? Cholesterol. GM1 ganglioside acts as receptor for the cholera toxin. Cholera toxin acts via the cyclic AMP. What kind of diarrhea is going to be seen? Rice watery diarrhea. How many types of phospholipids do you know? There are phospholipids like lecithin, sphingomyelin, phosphatidyl serine, and cardiolipin. Phosphatidyl serine flipping, PS flipping is seen in apoptosis. What is the marker of apoptosis? Annexin 5. Cardiolipin, where is it present? In the mitochondria of the heart. Anti-cardiolipin antibodies are seen in syphilis. Syphilis is a sexually transmitted disease because of Treponema pallidum, a spirochete bacteria. Next, Apla syndrome is an autoimmune disorder, mainly seen in females, causing recurrent abortions in the second trimester. Okay, done. With this, what we have completed? We have completed the different types of lipids in the cell membrane. Next, we'll discuss about the cytoskeleton topic. Hope the video is helpful. Thank you.